Okay, so good morning, everyone. My name is Mariam Montenegro, and today I'll be presenting research on cystatin, specifically focusing on designing immunogenic oligopeptides from nematode cystatins in silico. So this work was conducted in collaboration with Marcela Oces Garay, Rosales Rios Martinez, under the supervision of Dr. Dariluz Mendoza. Okay, so before we talk a little bit about the research, it's important to talk a little bit about its background. So why cystatins? Cystatins are eukaryotic proteins involved in regulating proteolysis and immune responses, playing an important role against the defense of pests and parasites. So recent studies have shown that they have a potential capacity in treating diseases like asthma, rheumatoid arthritis, diabetes mellitus, and some types of cancer. So conventional biochemical studies of cystatins rely on molecular techniques, which are often very complex and costly, which require specialized lab settings. Also, the extraction of native proteins can risk denaturation and increase costs. Fortunately, advances in genomics and proteomics alongside with in silico modeling provides faster identification of peptides and other chemical compounds. Additionally, the search for a certain target or therapeutic agent in bioinformatic databases is a strategy that allows identifying and designing molecules with a high probability of being biological active and with the facility of being able to be synthesized in practice. So to carry out this research, we began with the in silico design of the peptide. So this included the search of the cystatin sequence in the biological databases, the prediction of B cell epitopes, and the analysis of the physical, chemical, and structural properties of candidate peptides. Also, we did the molecular modeling, which include the designing of the 3D structure and the validation of the model. And finally, the molecular docking, in which we wanted to see if the peptide was able to fulfill its natural function, which is to inhibit cysteine protease. So in this case, we use environmental allergens to try to determine the type of interaction. So when it came to the experimental part, we used the services of company Select Chemicals for the synthesis of the peptides. So they use the solid phase synthesis method, and with this synthetic peptide, we immunize Western rats. So the goal was to generate a specific anti-cystatin IgG polyclonal antibodies. So following this immunization, we collected the serum sample from the rats and performed an ELISA assay in order to confirm the presence and specificity of the antibodies. And finally, we utilize these antibodies in order to investigate native cystatins in whole body extracts of the nematode Ascaris lumbricoides. Okay, so first, like I said before, we use biological databases. In this case, we use UniProt and GenBank, which is from the NCBI. And we use the search under the Nematode phylum. So 20 sequences were selected with that had words like cystatin, cysteine protease inhibitors, CPI, L-cystatin, etc. So once we selected this sequence, we use T-Coffee for the multiple sequence alignment. So as you can see in the slide, the color scheme highlights the amino acids according to their percentage of conservation. And in this case, we can see 4 highly conserved regions, which are N-terminal glycine at position 161. We have glutamine, valine, aline, glycine motif in positions between positions 208 and 212. We have a disulfide bridge of between the cysteines 226 and 241, and we have a proline tryptophan site at positions 160 and 161. So this is important because these regions are known to be highly conserved in nematode type 1 cystatins and to serve important functions for their interaction with cysteine protease. So for the design of the B cell epitopes, the consensus sequence was entered into the antibody epitope prediction program from the IEDB, where 5 peptides were obtained. So thanks to the optimum antigen software from the company GenScript, we managed to find out that the peptide with the most better properties of antigenicity and facility of synthesis is the one shown here. And also, through this software, we determined that it was the one with the best probability of acting as an antigenic determinant in the murine model. So for the molecular modeling of the immunogenic peptide, which we call NCIS01, we use the software PECFUL based on the admission method. This means assuming there's no homologous template in the biological databases. So this software basically ranks the models it determines based on free energy, where the lowest free energy determines the most probable confirmation. So in this case, we use that criterion to show the model. Additionally, we wanted to validate if this model was probable, and for that, we used the Ramachandran plot. So the results of this analysis show that the total percentage of the residue was in the most favored areas, were almost 100%, thus validating our model. Because this plot principle is based on the fact that more than 90% of its residue must be in the most favored areas, which are the ones shown in red and yellow. Additionally, we did the molecular modeling. Like I said before, we used the cysteine protease of environmental allergens, and results show better results when it came to the allergen of blood T1, which is from the storage dust mite. It had an binding energy of around minus 6.6 kilocalories per mole. We also wanted to see if this type of affinity was through a recognizable binding pocket, and for that we use Protein Plus. The molecular docking was done using Autodoc-Bina, and we also wanted to see what type of interaction was going on, and for that we use BioVira Discovery Studio. And as you can see here in the slide, there's a high number of hydrogen bonds and electrostatic bonds like salt bridges, which means that this suggests that the peptide has a strong recognition activity and it might have an inhibitory activity. Of course, further studies need to be done to confirm this, but this is a good preliminary analysis. So it came to the synthesis and the production of antibodies. Like I said before, it was through the services of Silico Chemicals, which they provide 10 milligrams of the peptide with a molecular mass of around 1562.8 Dalton and a purity of around 88.2%. So figures 6A shows the corresponds to the electrophoretic profile of the sera of the immunized rats from the peptide. So in this case, we had that the concentrations of total proteins in the control group were around 6.13 and 40.99 milligrams per millimeter, while in the control rat was around 25.9 milligrams per millimeter. Additionally, we have the figure 6B and 6C basically show the purification process of the IgG polyclonal antibodies. In this case, with figure C, we have that the column 9 is the one that shows the IgG antibodies that were eluted from the column. So when it came to the antibody titration and one-star blood, we have that the components of the antibody titration are here showing figure 7. And we managed to see that 3 of the rats that were immunized with the peptide produced sera with good titers against a total protein extract of the Nematode Ascaris lumbricoides, even at the lowest dilution tested. So the capacity of these sera to recognize Ascaris lumbricoides were actually confirmed or determined through the Western blot analysis. which is shown here in figure A. So here we managed to detect 5 major electrophoretic bands, one of which shown shows a molecular size close to the native cystatin of the parasite, which is around 16 kilodalton. So in summarize, we managed to demonstrate that immunogenic synthetic peptide designed in silico can generate a specific antibodies against nematode cystatins. So this approach offers a promising alternative to traditional methods, providing a tool for developing immunoregulatory drugs and potential vaccines. So future research will be focused on confirming the identity of the 16 kilodalton protein from the Ascaris lumbricoides, which were recognized by the antibodies, and further assessing their therapeutic potential. Thank you very much. Is there any questions or comments?